everyone is there.
GoPro, turn on.
All right. I think we're all set. I'll just change my view. Okay. So, good morning, everyone. Welcome po. Good morning. It's a beautiful Saturday morning for another webinar from the Filipino Science Hub. Magandang umaga po sa lahat ng participants natin here on Zoom and also from YouTube. I think we're now live through YouTube live stream. And welcome po sa to another special topic offered by the Filipino Science Hub Research University. What's new in the world of fungi, taxonomy, and nomenclature? So today we are joined by our guest speaker, Dr. Connie Cañete-Gibas, uh, to talk about all about fungi, its taxonomy, classification, and nomenclature. So, but before anything else, we would like to share uh, some some requests. We would like to kindly request everybody to uh, turn off their microphones. Kindly mute, please, please. Uh, let's show some love to our guest speaker by not interrupting her in the middle of the talk, and also please kindly turn off your videos so that we don't lose bandwidth. Mamaya po you have the option to turn on your videos and microphones to pose a question or a comment live uh, pagkatapos na po nung presentation ni Ma'am Connie. Okay? And please no recording of the session because the full video recording will be posted on YouTube and it's readily accessible right after the webinar. Okay? All right. So sa mga nagtatanong po, we will all of our web events come with a certificate of participation, and we do that through a Google form, the link of which we will post here on the Zoom chat box and also on the YouTube live comments. And right after the webinar, yung pong link, Google form link, is posted on the video description in YouTube of this event. Okay? And in order to obtain your certificates of participation, you have to be a subscriber of Fieldside Hub's official YouTube channel and the Facebook group. Okay? Okay, so as we have done in the previous webinars, we would like to reintroduce ourselves to those who are joining us for the first time. We are the Filipino Science Hub, and here to uh, give you our introduction is our CEO and founder, Dr. Jeffrey Bonkin, who is joining us from Texas, USA. Hello, Doc Jeff. Good morning.
Hello, good morning, JP.
Okay,
Magandang
I'll give
umaga.
the screen to you now.
Okay, magandang umaga po sa inyong lahat. Ako po gaya na po sinabi ni JP, ako po si Jeffrey Bunkin, ang founder ng Filipino Science Hub. So, ako po ngayon ay nandito sa Houston, Texas. Ako po isang Filipino scientist. At actually, ang ating pong speaker ngayon ay napakahaba po ng history ng aming pinagsamahan, but I will introduce her later on. But for now, I would like to introduce to you Filipino Science Hub. So, Filipino Science Hub is an online platform that we founded back in 2012, and its main intention is to actually promote the cultures of STEM education and research among students and teachers in the Philippines. So at the moment, Filipino Science Hub has 7 core team members, and so we have Dindiyu, also is based in Kentucky here in the US. She's also a Filipino scientist trained in chemistry. We have John Marty Mateo, who's a scientist at the Institute of Plant Breeding in UP Los Baños. And then the JP Onya, the one who opened the session, is currently a researcher in electrocatalysis in Finland. Professor Medjon Aguila, who's a professor of chemistry at the University of the Philippines, Los Baños. We also have Janice Averelia, who's a Filipino scientist like in food sciences, and she's based in Kansas right now. And then last but not the least, Ralph Lauren Alomia, who's also an assistant professor of chemistry in UP Los Baños. So, what we're trying to work hard towards is at Filipino Science Hub is to actually create a new culture of STEM among Filipino sciences as science enthusiasts, and like the younger generations. So, and we approach this through two very important elements of launching a culture, and that is to actually promote the foundations, or to strengthen the foundation of STEM literacy among students and teachers. So dito po, meron kami programa through Fieldside Ed that aims to deliver materials that would actually help empower our educators. So yun po yung mga ginagawa namin sa Fieldside Education. But launching a culture does not stop at educating you. You have to be able to practice or to apply your knowledge in order to innovate. That's actually how you relaunch the STEM culture, and we do that by our program at Fieldside Research University. So what we do at Fieldside Research University is that we provide trainings on research fundamentals, and we also bring in yung mga Pinoy scientists po natin at mga Pinoy career practitioners, and we're bringing you some more of a practical sense of the science concepts that you learn in school. So ito po yung aming strategies sa Fieldside Research University, and then later on our team members will actually introduce to you the specific programs under these components. Okay, and then the vision that we have is that so for example, for Fieldside education, the focus here is to actually again empower our educators. So if you could imagine, if we have 30,000 followers on Facebook, if those are actually all educators, and each of these educators would teach for about like 30 years, through the course of their career, and for each year they actually get to influence 30 students at a time. You're looking at about like 27 million students impacted over the entire course of our academic frontliners career. So ito po yung vision namin sa Fieldside Hub. And if you notice, like if our community keeps increasing, so you could just like imagine this number growing much bigger. So yun po yung aming goal dito sa Filipino Science Hub, and in particular through Filipino Science Hub Education. Para po sa kabatiran ng ating mga guro, we started our online events last year focusing on teacher training, because at the onslaught of the pandemic, we had to make sure na ready po ang ating mga teachers. So we started with teaching webinar. So we had actually shared strategies on how to transition to online or to distance teaching, and we actually invited a number of seasoned Filipino academicians from different parts of the world. And sila po yung nag-put together ng training courses na to. So these are actually all still accessible on our website and on our YouTube channel. So if you need some guidance as to how to transition into distance teaching and, adapt effective ways of, teaching online, these are things that you could actually check out. But then it actually doesn't stop there because we want to make sure that we also get to deliver STEM contents that the teachers can use to more effectively teach foundation areas in STEM. So like very recently, we actually launched Felsyab Ed's modules program. So essentially, on a daily basis, we have been rolling out teaching modules and practice problem sets in the areas of biology, chemistry, and physics. So ito po, these are contents that we put together in accordance to the Dept standards of specifically when it comes to the MELCs, the most essential learning competencies that the students should actually take away from each course. And specifically, we actually and for this we recruited 3 staff members who are actually curating all these modules, like on a weekly basis. And these are Bachelor of Science and in Math and Science teaching majors from the University of the Philippines. So magagaling pong mga bata. So these modules contain teaching guides. They also contain prescribed class activities, visual aids that the teachers can use, and most importantly, problem sets that not only the teachers can use, but also magagamit ng mga estudyante na mag-practice po para mas ma-improve yung understanding nila nung subject matter. So these are things that you can access through our online platform, and later on, like JP will actually show you where like all the platforms like in which we're actually present. All right, take it away, JP.
Okay. So as Doc Jeffrey introduced, we also have our campaign, Fieldside Research University, which is a webinar series focused on scientific research and its different aspects. So our aim at Fieldside Research University is to put into practice all of the information and the knowledge that we impart here at Fieldside Hub into a capstone project, which is basically your science investigatory projects or college undergraduate thesis. And here in Fieldside Research University, we will share some of the best practices in terms of carrying out a research project, and we will share our own research experiences and some tips and advices in going about scientific research. Next slide. Okay, so Fieldside Research University is composed of 8 mandatory courses, and we covered the entire range of a research endeavor, starting with the ideation, how do you do the literature review, experimental design, all the way to poster and conference presentation. So, 8 po ito, 8 mandatory courses, and we are now halfway through these courses. So the next one would be on April 17,
Yep.
which is all about data analysis and statistical design of experiments.
Yep, and JP, we have almost 1,000 students already taking up this course, this program.
Yes. So imagine the impact it's gonna make among our teachers and students. Yep. Okay, next slide. Okay, so we devised a Google Classroom for Fieldside Research University, and all of the details is summarized in our launch last January. The link of which I will post later dito po sa chat box, and you will also receive the details when we send out the certificates of participation later. Okay, so dun po sa Google Classroom namin, we already have more than almost 1,000 students, and baka mag-open po kami ng panibago. So we'll keep you informed on that. Okay? So ito po yung Fieldside Hub platforms. We have various channels to which we can reach everyone. So the main platform that we have is our website, www.filsihub.com, where we share all the webinars, tutorials, modules, virtual labs, and some other features. So kindly check it out, www.filsihub.com. And ayan, marami po, lahat po ng offering namin ay nandun. So kindly just like explore and browse through every section of that. And we're also ever present on Facebook, where we have more than 29,000 followers, and on YouTube. So you can just type in Philsci Hub and lalabas na po yan. And we're also even present on TikTok to reach out to our younger audiences. Okay, the students. Ayan, so to those who just joined in, we are on our way to presenting our special topic for today: what's new in the world of fungi, taxonomy and nomenclature. And to um, introduce our guest speaker, I give you back Doc Jeff Bunkin to introduce our speaker, Dr. Connie Cañete-Gibas.
Okay, back to me. Okay, so I will actually I insisted that that I introduce our speaker for today, not only because she's a world-class Filipina in the sciences, or specifically in her area, but more so because she's like a family to me. So nag-meet po kami ng speaker natin when I was still a graduate student in Canada back. And I think when was that, Tita? 2009, 2010. So about 10 to 11 years ago, like doon kami nag-meet sa Canada pa. Parehas kami giniginaw, and then para kaming ampon ni Tita. So we were grad students back then, and she would actually feed us on weekends. So she would invite us over. She's very nurturing in that way, and she's equally nurturing when it comes to like the passion of Filipino students, of the younger Filipino generation when it comes to the sciences. That's the reason why she actually also volunteered to do this for us, and also like a couple more of very nice offerings later on. So but this is gonna this please consider this session as our introduction to the Philsci Hub community. Okay. To begin with her introduction, so siya po ay Pilipina. She's actually from the Visayan region. So our speaker for today, she did her undergrad in medical technology in Saliman University. So hindi na natin ilalagay yung year kasi bata pa tayong lahat dito, no, Tita. And then after that, she actually took up her master's degree in veterinary medicine. Specifically veterinary microbiology as a specialization at UP Los Baños. And dun siguro na-meet ni Tita yung love of her life na si Tito. And then after that, she actually took up her doctorate degree in biological sciences, specializing in fungal systematics at the University of Alberta in Alberta, Canada. So dun din po ako nag-aral ng PhD ko. And then she has like a very storied career. So she worked as a university researcher for the Philippine National Collection of Microorganisms in Biotech. It's actually a national research facility based in Los Baños, Laguna. She also was a research fellow at the Japan Collection of Microorganisms at the Riken Wakoshi, Japan, and Japan, and at the University of Tokyo. So University of Tokyo is actually one of the best universities in the world, for your information. After her PhD, she also worked as a curator for the University of Alberta Microfungus collection and herbarium in Canada. And then at the moment, she's actually working as a clinical mycologist and clinical research project manager at the Fungus Testing Laboratory at the Department of Pathology and Laboratory Medicine at the Long School of Medicine at the University of Texas Health at San Antonio, Texas. So medyo may pagkatang twister po yun. Sobrang haba ng mga credentials ni Tita, nakakatuyo siya ng lalamunan. But that's how good she is. But we actually don't stop there. She also has discovered like new families and orders of fungi, and like a great number of new species, and yun yung mga i-share niya sa atin ngayong araw na ito. So para dun sa mga estudyanteng nasa inyo, so nasa Pilipinas, so makinig po kayo sa storya ng buhay at ng career ng ating speaker, kasi like this is actually one that will inspire a great many, and most especially like young Filipino women out young female Filipinas out there who are actually interested in forging a career in the sciences. So, ladies and everyone, ladies and gentlemen, it's my great honor to introduce to you my very dear tita, Dr. Connie Gibas, Dr. Connie Cañete-Gibas. Tita, take it away.
Okay. Can you hear me?
Yes.
Fine?
Yes, loud and clear, tita.
Yes, loud and clear. Hello, everybody. Thank you so much,
Good morning,
Dr. Jeff, for the very kind introduction. And thank you, the Fieldside team. This is a very, very wonderful platform. So good morning, everyone in the Philippines. To Dean Rosemary Gutierrez and company sa UP Baguio, Dr. Dalmacio, Dr. Lantican, and Director Marian De Leon, Dr. Monsalud, dyan sa UPLB.
Are they tuned in right now, tita?
Lahat, ha?
Are they tuned in right now?
Yeah. Those are some familiar names. I have to
They
say.
are.
Okay.
And Dr. Oxy Siringan, and colleagues at UP Diliman, Dr. Aya Torres, Dr. Jason and Ruth Cantera sa Washington, D.C. Good evening diha sa inyo. And Ms. Pascual sa San Diego. And lahat sa Dr. Parco sa Silliman University. Mayong buntag, Dr. Parco, Dr. Sok, and sa Visayas and Mindanao, mayong buntag po ninyong tanan din ha. Mayong paniud to, di ay. Okay, so before I go to my main topic, let me tell you a bit about how I became a clinical microbiologist, mycologist. Kasi galing yan sa microbiologist, tas naging mycologist. Okay, so next slide. So how did I get there? Do you remember when you were little, everybody would ask you, anong gusto mo paglaki mo? Ang iba sabi, gusto ko maging pulis. Yung iba, maging teacher. Ako talaga gusto ko maging doktor. Kaya lahat na lang, tinatawag ko Dr. Laway nung maliit pa ako. Kasi lahat na lang may sugat, nilalagyan ko ng laway para, kasi doktor daw ako eh. So anyway, I grew up in a very small barrio sa Fatima, Santa Catalina, Negros Oriental, sa Bisaya. Tapos, this barrio is really small, like less than 5,000 population. Kaya, imagine mo, promdi talaga ako. Yan, bariotic talaga ako. Di ko alam kung paano yung sa city, di ko alam yung TV. Lagi mga cousins ko, nanood ka ba ng Chiti Bang Bang? Ano yun? Kasi wala kaming TV sa barrio, di ba? So anyway, the point was, nasa barrio ako, tapos nagpunta ako sa city for high school. So ma-imagine mo yung culture shock. Pagdating ko sa high school, tinatanong ako, sisimba ka ba? Kasi lagi ako pang-simba ng damit kasi wala pa akong uniform noon. So ganyan ang shock. So anyway, nasanay ako dun sa Dumaguete. So I went to school, sa high school, sa Silliman University High School, which is a very good high school. Yeah. And yun ang foundation ng aking education talaga. So that's where it started. Because I read a lot of books. The library is very rich. So anyway, after high school, so sa fourth year kami, so hindi pa ako nag-medtech. Narinig ko yung mga barkada ko, sabi, pupunta sila sa CEBU, yung gagraduate na kami ng high school. Sabi, where are you going? Tapos sabi, punta kami CEBU. Sama ako. Bakit? Anong gagawin nyo doon? Kukuha pala sila ng upkat. Hindi ko alam. So anyway, sabi ko, ay, sama ako. Tapos nagpaalam ako sa tatay ko. Sabi ko, pa, pupunta kami mag-field trip. Kasi hindi ko sinabing kukuha kami ng exam. Kasi hindi ako papayagan. Kasi hindi naman siya papayag na mag-aaral ako sa ibang lugar. So anyway, I went to Cebu and I wrote sa upkat. Tapos nung summer na yon, my father received a letter galing sa Diliman. Sabi, nakapasok daw ako sa zoology, sa degree program. Tapos sabi niya, hindi ka pupunta. Sabi ko, paano na yon ang aking plan? Di ako magiging doktor kasi doon ako sa zoo, yun ang aking pre-med. Sabi ko, plan B. Nag-medtech na lang ako sa Silliman kasi gagraduate ako ng medtech. pwede pa ako magtrabaho kahit matapos ako as a pre-med. Tapos pagkatapos ng medtech, naghanap ako ng trabaho. Alam mo ba nung time na yon, after I finished medtech, merong rural medtech na program sa gobyerno. So they're supposed to hire yung mga newly graduates. Punta ako dun sa aming medical officer. Sabi ko, mag-a-apply ako ng rural medtech kasi I know dun sa town namin meron doon clinic. Alam mo sinabi, you have to have a letter from your mayor, your governor, your congressman. Of course, nahirap yun, di ba? Ayaw ng tatay ko ng ganon. Di ako napunta sa mayor. Anong ginawa? Doon sa isang, alam mo yung wrapper ng sigarilyo? Doon nilagay yung note. So, offended talaga ako. Ano ba yan? Bakit ganon? So, hindi na ako nag-apply. So, that summer, nagpunta akong Cebu. Sabi ko, ma-mark time ako sa Cebu kasi hindi ko pa alam kung anong gagawin ko. Nag-apply ako ng trabaho. Nakuha ako doon, sales rep. Alam mo ba yung, I'm gonna knock on your door, ring on your bell. Tap on your window too. Yun yung naging trabaho ko. Nagtinda ako ng electrolock. Tapos 2 months ako nagtrabaho. Wala akong benta. Kaya tanggal ako sa trabaho kasi hanggang kahit basta pag 2 months wala kang benta, hindi ka pwede na magtrabaho. Anyway, balik ako sa bahay. Sabi ko, punta na nga ako sa Manila. I will try my luck there. Tapos nag-apply ako, natanggap ako research assistant sa College of Vet Med sa Diliman. So, yung sa vaccines lab. So, dun, tsaka lab ko yun. Gusto ko niyang research eh. So, after a few months, na-relocate yung vet med sa UP Los Baños. I had to go with them kasi wala akong trabaho kung hindi ako sasama. Anyway, nagpunta na ako sa Los Baños. Ang lab ng vaccines, nilagay sa biotech noon, sa National Institutes of Biotechnology and Molecular Biology. Tapos, from there, yung position ko, imbis na research assistant, naging lab tech. So, yun ang kanilang pangalan. So, sabi ko, after 5 years, sabi ko, I don't want to be a lab tech forever. Sabi ko, babalik ako sa school. So, mag-grad school uli ako. So, I was thinking, so that time, yung mga lab techs, hindi pinapayagan mag-aral. Kasi research assistant pataas. Kasi parang walang kayong mag-aral, lab tech ang kayo eh. So anyway, nag-petition ako kay Dr. Padulina. Sabi ko, kinausap ko siya, gusto ko mag-aral. Kasi gusto ko mag-advance professionally. Sabi niya, okay. Kasi he was very good. Papayag ako, pero may condition. Kailangan matataas ang grado mo. Sabi ko, opo, sir. Kaya ko yun. Sabi ko. So, yeah, and then I enrolled. So, I got admitted to the program sa Master of Science in Veterinary Medicine. Tapos, ang specialization ko ay microbiology. But at that time, when you're working full-time, you can only do 6 units at a time. So, it took forever bago matapos yung MS ko. So, in between that, natanggap ako sa UNESCO program, postgraduate program in microbiology. So, this was my watershed moment. Ito yung turning point ng buhay ko. Kasi when I was there sa University of Tokyo, yung pinag-aralan ko doon, so I was working on the Taxonomy and Nomenclature of Aspergillus. So that was my first exposure to taxonomy. And I found that I really loved it. I loved it. Sabi ko, ay naku, dito ako mag-uusbong yung aking whatever it was. Anyway, so sabi ko, sige, ito na ang gusto ko. Nalimutan ko na yan. Gusto ko pa rin yung maging doctor of medicine. Pero alam ko, hindi na yun eh. Hindi na yun realistic. So, doon sa University of Tokyo, I was studying the classification of Aspergillus, the genus Aspergillus. So what I was trying to do is, I was trying to look for taxonomic characters. Yung pag sinabi mong taxonomic characters, yun yung characteristic ng fungus, ng isang species na magiging you are able to differentiate from the other species na closely related. So enzyme patterns, DNA sequencing, DNA homology using radioisotope. Yun ang inaral ko lahat yun sa University of Tokyo. Biro mong training yun, ang galing. 1 year lang yun, nagawa namin lahat yun. Nagawa ko yan lahat. So after that, sabi ng aking, umuwi na ako para tapusin yung MS ko. So I went back to Los Baños. Tapos tinapos ko yung MS ko sa VetMed. Tapos that year, there was a meeting, international culture collection meeting. So doon naman yung sa Yogyakarta sa Indonesia. Na-meet ko naman yung director ng Japan Collection of Microorganisms. Tapos sabi niya, Connie-san, gusto mo ba magtrabaho sa JCM? Sabi ko, pwede. Sabi ko, di pinapunta niya ako doon for 1 year. Pinagtrabaho niya ako ng isang project. So doon ko na-discover yung 3 bagong species ng yeast. So that was the beginning. 1998 yon. That was the beginning na nagsimula akong mag-discover ng fungi. So anyway, nung nandun ako that time, na-meet ko naman yung magiging supervisor ko sa U of A, si Professor Sigler. Pero mo yung mga pasikot-sikot na hindi mo naman plano, pero there are events na mag-lead sa isa to another. Diba? So, huwag kang ma-frustrate kung hindi natuloy yung plano mo. Kasi meron niyang pupuntahan.
Redirection.
So, nung di sinabi ko, pwede ba akong maging student mo, PhD student? Sabi niya, okay, sige. Kasi nalaman ko siya, she was one of the who's who sa mycology. So, gusto ko mag-train under her kasi, di ba, dun mo doon ka sa magaling para ang galing din ng training mo. So, noon, nakapunta ako ngayon sa University of Alberta. So, pagdating ko doon na talaga nag-immersion na talaga ako ng fungal systematics. So from there, dapat pupunta akong CDC para mag-postdoc. Tapos sabi niya, no, don't go to CDC kasi maliit ang bayad doon. Dito ka na lang, magtrabaho ka na lang, i-hire kita as associate curator kasi siya yung curator noon. Yung University of Alberta Microfungus Collection, it was the 3rd largest in Canada noong time na yon. Tapos the 8th largest in the world. So, okay, yes, it was a gold mine. Kung gusto mo talagang mag-study ng fungi, you have 12,000 strains in the collection. That's a lot. You can screen a lot of fungi. So doon naging associate curator ako. Tapos in 2012, nag-retire siya, naging curator ako doon. But soon after, kasi while being there, lagi kaming may collaboration sa mga labs sa US, sa Europe. So, yung director ng fungus testing lab, 2012 pa yon, nag-approach na siya sa akin. Sabi niya, do you want to join us sa fungus testing lab sa Texas? Sabi ko, isipin ko pa. Tapos, tinanong na naman niya ako the next year. Sabi ko, sige. So, yun, sabi niya, okay, kasi sponsor nila ako papunta dito sa US. So that's how I got to be here in the US. So I was dito sa University of Texas as a clinical mycologist and a clinical research project manager dito. So yun, ang kwento ko. Parang lahat na lang nangyayari na hindi mo naman finlano talaga. Pero there were very pleasant surprises talaga.
Yep. And so yun, message sa mga bata, no? So kung baka may plano kayo, huwag kayong ma-frustrate.
Huwag kayong ma-frustrate. Ang kung ano yung gusto nyong maging trabaho, doon you work for it na lang. Kasi sa akin, ang dami kong na-waste na time. Kasi pasikot-sikot pa ako. Pero kasi hindi ko naman alam talaga na I would come to love yung mycology. Mm-hmm. Okay, so that was my story. So if you want to be a mycologist, ito yung pwede mong gawin. Ang minimum education required is that you have to have a degree in microbiology, or allied field na merong substantial coursework sa microbiology. Kasi yun ang training mo, yun ang foundation mo. Tapos, after nung bachelor's, magti-train ka ulit sa yung postgraduate studies. Kasi doon, magiging specialized ka na. Tapos, mag-postdoc ka. Ang postdoc mo, pwedeng clinical mycology, medical microbiology, or infectious diseases. Tapos, kung gusto mong mag-advance, halimbawa, nagtatrabaho ka na sa isang lab, gusto mong maging director sa isang lab, kumuha ka ng certification. So either mag-write ka ng exam from the American Society of Clinical Path or the American Board of Clinical Microbiology. Okay, so next slide, please. So ito yung mga certifications. So if you want to be a technologist in microbiology, dun ka sa ASCP, Molecular Diagnostics Fellow, pwede kang kumuha ng exam sa American Board of Clinical Chemistry. Tapos, pag gusto mo maging diplomate, doon ka sa American Board of Medical Microbiology. So those are very good. Okay? So now, we're going to go to our main topic. So what's new in the world? The next one, JP. You still have much room for improvement, okay?
Mushroom.
Yes. Okay, let's go to the next slide. Okay, so my objectives, the objectives of this talk is to learn some basic morphology of fungi. So the basic morphology I'm talking about will be relevant to what I'm going to talk about on nomenclature and taxonomy. Kasi talking about the morphology of fungi, it will take us the whole year. Ilang semesters yan bago matapos. Hindi lang isang oras. And then, so another objective is to become aware of the changes in fungal taxonomy and nomenclature and learn how to apply new rules in fungal nomenclature. Kasi marami ng bago ngayon. Next, please. Okay, so ito yung outline. So I'm gonna give you a brief background on fungal biology. Yung relevant lang sa talk na to. And then, why there is a dual nomenclature of fungi? Bakit dalawang name ng isang fungus? Tapos, why there was a switch to unitary nomenclature? What happened there? And then, I'll give you some updates in fungal taxonomy a little bit. And then, I will teach you how, or I will show you how you establish a new higher order classification. Like, if you have a new order, you need to do a family also. And how you can also describe new species. Okay? But first thing first, I'm sure some of you have read this when you come across articles on fungi, or you see MOULD versus MOLD. Well, MOULD is a British English preference, and then MOLD is American. Ang sabi kasi nila, kung MOLD, yun yung mold na nag-shape.
Yeah.
Hindi naman kasi every time you talk about it, may context na fungus. Hindi naman lang, basta mold lang pinag-uusapan. But anyway, sa Canada kasi, MOULD. Pagdating dito,
British
MOLD. Hindi, sometimes, hindi mo alam kung anong gagamitin mo. Yeah, so, ganun yun. Okay. So what is taxonomy? So it is a system of naming and organizing things, especially plants, fungi, animals, and other organisms that share similar qualities. Yun yung keyword doon, yung pare-pareho sila itsura. Kinogroup mo together. So biro mo yon, pareho tayong brown, together tayo, related tayo. Kayo mga puti, related kayo. Kayong mga maliliit, related kayo. Pero do you realize how plastic that is? Basta lang magkapareho ang itsura, pwede nang i-classify together. So that was the old traditional way. Okay, next slide, please. So then what is nomenclature? So nomenclature is the devising and choosing of names for things, especially in a science or other discipline. So example, yung Linnaean system ng zoological nomenclature. Yun yung nakagisna na natin, yung Oryza sativa Linn. Linnaeus yun, di ba, si Linn? So yun yung authority ng name na yon. So, yun yung name ng rice, di ba? And then there's the nomenclature of chemical compounds. I'm sure you mga chemistry guys dyan, yun ang ginagamit nyo, di ba? So, it makes you, there's a system. So, hindi ka lang pwedeng magbigay ng, oy, ang pangalan mo si Connie. Bakit? Bakit ako naging Connie, di ba? Meron talagang system, may regulation, meron siyang sinusunod na criteria. Ganun din sa inyo sa nomenclature ng chemical compounds, right? Okay. So, that's what it is. Meron yung criteria bago mo choose yung names na yon. Okay, so let's talk about the kingdom fungi. The kingdom fungi is also called kingdom Eumycota. So Eumycota means true fungi, where mycota came from the word mycos, which is Greek for fungus. So fungus is filamentous or unicellular, and they are heterotrophic, meaning that they can get their energy from organic compounds, and they have no chlorophyll. So mga saprobes yan sila. So, well, another thing, I always see that in articles, they say saprophytic. That is wrong. You just say saprobic. The correct term is saprobic. So hindi saprophytic or saprophytes. Say that tends to pertain to trees, right? So hindi yan ganyan. Dapat saprobic. Anyway, nag-detour na naman ako. So fungi has rigid cell walls, and they contain chitin and beta glucan. So yun ang nagdi-differentiate sa kanila from bacteria. Okay. So let's start with knowing the fungus. So there are two fungal forms. So there's the yeast form and the mold form. Now, I know most of you know this, but nakikita ko lagi ito sa mga clients namin, sa mga clinicians na, mga techs na tumatawag sa amin. Sasabihin sa akin, no, I did not send you a mold. I sent you, no, ang sabi niya, no, I did not send you a fungus. I sent you a yeast. I go, well, fungus and yeast is a fungus too, sabi kong gano'n. Kasi hindi nila alam yon. Kaya yun, nilagay ko siya dito para alam nyo lahat na a yeast is a fungus too. It's just the unicellular form. So pareho sila. So don't be confused. Okay? Then, next one, please. So a few terms. Kasi ito relevant to sa ating talk ngayon. So one is like, there are fungi that are polymorphic. Polymorphic fungi means that there are those that have more than one independent stage in the life cycle. So polymorphic, marami siyang morphologies. And then we have the holomorph, which has all the stages of the life cycle of a fungus. And we have the teleomorph, which is the sexual state. The anamorph is the asexual state. So, tagin natin, babanggitin yung teleomorph, anamorph in the next slide. So please, teleomorph, sexual, anamorph, asexual. Teleomorph, sexual, anamorph, asexual. Okay? And then the two of them together, it becomes a holomorph. So yun. So again, the life history of a fungus. The next one, please, JP. So this is the life history of a fungus. So they have the anamorph and the teleomorph, but they can occur at the same time. Sometimes they occur independently from each other or at the same time. So if they occur at the same time, yun ang nagiging holomorph siya. So get nyo na? Okay? Kasi I know fungi, when I talk about fungi, it's very jargony. So some terms that are very familiar to us, but we don't really know when it comes to fungus, the meaning is different. Okay? It's the same with chemistry. There are a lot of jargons that I don't understand when they're talking in chemistry, but you're familiar with the words, so bear with me, okay? So I would like to give you examples of polymorphic fungi. So here's a species called Blastomyces dermatitidis. So they have the mycelial or the hyphal stage, and then they have the yeast stage, which is called the BBB. So broad-based budding yeast. Sometimes they're in tissue, but sometimes they can occur in culture also. So they're together sometimes. So that's why Blastomyces is also called dimorphic because it can change into a yeast form in tissue, but also pleomorphic because they can occur together in culture in vitro. So on your plate, like you can see them when you make a mount. Okay, so another example of a polymorphic fungi is the Pseudolicheria boydii. Nice name, right? Baka mama pangalan nyo sa mga anak nyo yun, ha? Uy, Pseudolicheria!
Kind of a mouthful po.
Ha? It's a
Mouthful po siya, tita.
Baka po ano na lang, baka pina lang, tita.
Pwede rin. So, anyway, etong Pseudolicheria boydii, polymorphic fungi. So makikita mo meron siyang what we call the Grafium anamorph, which is a bunch of the Scedosporium anamorph. So if you look at the Scedosporium anamorph, it's just a single conidiophore with the conidia on the stalk. And then when they're bunched together, they become the Grafium anamorph. So when you see this in culture, do not panic because you don't have a contaminated culture. These are just polymorphic fungi having two morphologies together or two states. So kung dalawa ang anamorph mo, ang tawag doon, synanamorphs. So one or two. So marami siyang anamorph. So synanamorph siya. Okay? So that's another example of a polymorphic fungi. So then here are examples of the anamorphic fungi in the phylum Ascomycota. Now I'm showing you this so that when you're looking for this, when you're looking at your cultures, you will want to look for these structures because these are the ones that you would need for identification. Otherwise, there's nothing to go by kung wala kang makikitang ganito. Okay, so these are anamorphs or the asexual state of species in the phylum Ascomycota. So here's Cladosporium with the typical shield cell, and then there's the acrimonium with the sticky heads, and then you have the boat-shaped macroconidia in the fusarium, and then you have the arthroconidia of coccidioides, and you have alternaria and Aspergillus. So I'm sure most of you are familiar with these fungi. And another slide, next one, are examples of teleomorphs naman, mga sexual structures ng species in the phylum Ascomycota. So here, the first one is a Cleistothecium. So it's a structure that is round, but it's completely closed. Open lang yan kasi in-squish ko bago ko kinuha na ng picture. Yan yung bago kong discovery noong 2002. Anyway, so completely closed. Tapos yung perithecium, round din siya, pero meron siyang opening, as you see in that drawing. And then yung pseudothecium, yun yung ascoma na embedded siya sa tissue ng plant na ini-infect niya or in the tissue of a patient or sa yung may sakit na patient mo. So that's pseudothecium. So na-embed siya sa ilalim siya ng tissue ng plant or leaf. Thus we have the gymnothecium. Those are what we call the naked ascomata. Kasi you can see through them. So naked siya. And then you have the apothecium, which is what we call the cup fungi. Makikita mo yan sa field, sa mga woods. Na mga orange siya na gano'n. Cute. Para siyang mushroom, pero hindi siya mushroom. Okay. Next one, please. So here is an example of the teleomorph and anamorph together in one plate. So this is the Neosartoria anamorph. And then you have the Aspergillus so Neosartoria teleomorph and the Aspergillus anamorph. So makikita mo siya, meron yung mga round structures, hindi masyadong kita sa plate. But if you make mounts, this is what you see. Okay? So note here that the Neosartoria teleomorph is different from the name of the anamorph, which is Aspergillus. So yun, pag-uusapan natin yun sa next slides. Okay, here are examples, or an example of a teleomorph and anamorph in the phylum Basidiomycota. So alam mo yung Basidiomycota, yun yung mga mushrooms. So, makikita mo yung teleomorph niya kasi nakikita mo sa field, yung bracket fungi na nasa trees, nakikita mo yon, that's a teleomorph. And then yung mga kinakain nating mushrooms. Tapos yung puffball, that's a mushroom too. Kikita mo yon, pagka sabihin mo, pag kumulog, maglalabasan yung malalaking puffball. Sabi mo, tas iihian daw ng aso, merong mushrooms na lalabas. Oo, kasi merong mga tawag na gano'n, yung urea fungi. So, which is this Coprinellus radians. Nakikita mo yun sa woods. So, mismo, kung gusto mo mag-isolate niyan, maglagay ka ng urea. Tapos, babalik ka the next day, makikita mo may tutubo na doon. So, pwede ka na manguha ng sample na yon. So, anyway, ang makikita mo sa Basidiomycota, yung mushroom, pero yung anamorph niya, para lang siya yung mga anamorph ng Ascomycota. Katulad nitong Coprinellus at saka Phanerochaete. Okay? So now we go to the nomenclature of fungi. So why do we need, doon muna tayo, JP, sa first na yan. Kasi baka magulat kayo. Why do we need to name fungi? Alam nyo? So in one of my classes, yung mga estudyante ko ay mga medical residents, yung isa kasi pilosopo, sabi, bigyan mo na lang ng 1, 2, 3, 4, 5, 6. Yeah. Okay, pag sinabi kong fungi number 1, alam mo yung kung anong itsura nun? Hindi. Anong ginagawa ng fungi number 1? Di ba? Wala kang idea. It's a number. Pero pag sinabi mong penicillium, kung familiar ka sa penicillium, alam mo kung ano itsura kaagad. Alam mo kung anong ginagawa ng fungus na yon. So that's why we have to have a name for fungi. Because it evokes images of what the fungus looks like, and then it gives you an idea what the fungus is and what it can do, right? So fungi can be also our friend or our foe. So as a friend, it can help us bake the tallest bread. Like, alam niyo yung saying na bread is like a sun. It rises in the yeast and sets in the wake. Corny, no?
Ang dami mong baon, tita.
Ang dami kong baon. Busog na ako. Anyway, aside from baking, fungi can be used in fermentation. Alam na yon. Paborito yun ng mga estudyante. Beer, sake, ginagawa namin sa Tokyo dati. Whiskey, tapos food products like cheese and many others. And then, in medicine, yung meron kaming isolate noon sa University of Alberta, yung Tolipocladium inflatum, yun ang nag-produce ng cyclosporin. I'm not sure if you know, cyclosporin is an anti-rejection drug sa transplant patients. So it was a very, very good discovery. And then they're also useful as mycorrhizal fungi because they help plants get nutrition needed for growth. Kasi ganito lang yun, yung tree. Ang kanyang roots, pababa lang. Tapos kaunti lang, mga ilang metro lang. So kung halimbawa, ang nutrient niya malayo, nandun sa ilang meters away pa, ang fungi, that's why they're called mycorrhizal because yung rhizal, roots, yun ang pumupunta doon sa nutrients na yun, kukunin yun for the plant. And in the same time, bibigyan ngayon ng tree yung fungi ng food. So,
Okay.
mycorrhizal, symbiotic relationship.
Opo,
Yan ginagawa
mutualism.
ng fungi.
Para
So, meron kami yung project noon sa Smithsonian Institute, yung orchid mycorrhizal naman. But mamaya ko na lang yun ikukwento. Anyway, another one is as pest control. Merong isang fungus na talagang sumusuot siya dun sa mga insects, yung Bovaria. Makikita, meron pa ako nakita na yung, ang tawag namin ay marshmallow fungus. Kasi ang ginawa niya dun sa mga crickets, ang marshmallow cricket ang tawag namin. So isang cave, nakita namin, marshmallow, puno siya ng mycelia ng fungus. Namatay na yung kasi kinakain nila yung insect, yung fungus na yun. So they can be used as pest control.
And very specific po sila.
Yes, they are very specific. Yung Bovaria na species. So there are about 300 species that are known true pathogens. Alam mo yung true pathogens, yun yung pagka dumapo sa'yo yun, magkakasakit talaga yung nadapuan. Kasi kahit healthy ka, kahit hindi ka immune compromised, pag dumapo yung mga fungi na yun, it will cause disease. Pero meron yung opportunistic na pag hindi ka healthy, kung immune compromised ka, diyan ka magkakasakit. Pero otherwise, they're supposed to be friendly. Mga ubiquitous kasi yung fungi, you can find them everywhere. Pero wag ka, pag ikaw ay mahina ang resistensya, huwag ka na lang masyadong gumala doon sa mga lugar na yon kasi baka madali ng mga fungi na yon. Okay? So, I want to warn everyone, the following slides may contain images that may be disturbing.
So,
So,
kasi sabi kung kumakain po, ano?
Yes, viewer discretion is advised.
Yeah.
Okay, go ahead. Sorry. So again, why do we need to name fungi? This is one of the reasons why fungi should be correctly named. If you are familiar with the name of the fungi that cause these conditions, then you would have an idea what the fungus does and how to treat it. So what you see here, like those on the foot, it's called chromoblastomycosis, and it's caused by several species of dart fungi we call dematiaceous or phioid. An example is Madurella, and another species is Chimatospira grigia. Okay, so this fungus, kita mo yung mga lesions na yan, nagpuproduce yun ng fungus balls, maliliit na grains na nahuhulog na lang sa ground. Kaya pag naapakan mo yun, kakapit din sa'yo yun. Kaya nako, mag-ingat kayo dyan. So, ang hindi ko masyadong alam kung merong nagagawa ng study sa Philippines dyan. So, I'm sure marami tayong ganyan, pero kasi wala akong nakikitang reports. And then the other one on the arm, this is caused by Cladophilopora carionii. Yung mamang ito, I think he lost his limb. Kasi ni-resection nila, ni-surgery nila, tinanggal, naubos yung arm, pinutol na lang. So another species na Cladophilopora is yung Cladophilopora bantiana, which is neurotropic. Gusto niyang pumunta sa brain, so it's very deadly. So once na magkaroon ka ng ganon, nako, magdasal ka na lang kasi wala na mangyayari sa'yo. Okay? Another one. This one is also bad. Rhinocerebral mucormycosis. Kasi ang gumagawa nito, yun yung mga sugar fungi. Nakikita mo to sa bread, sa iyong refrigerator. Pero kasi yung may mga conditions na mga underlying conditions ng mga tao, they will get this kasi opportunistic like rhizopus mucor. So kita mo yan, pagka-admit pa lang niya, nangingitim pa lang yung kanyang pisngi. Pero after 12 hours, tingnan mo ang necrosis. Grabe na. Meron pang isang picture talaga na ubos talaga yung mukha. So hindi ko na lang pinakita kasi grabe talaga, hindi mo malilimutan. Baka kailangan mo lang pumunta sa psychiatrist para No. So, yun ang mag-ingat talaga tayo. Kung meron tayong nakikitang mold sa bahay, huwag mo na lang yun, ay wala lang yan. Kasi hindi mo na alam ngayon ang panahon. Kasi marami ng, they're becoming very refractory, meaning that marami silang resistant na sa mga drugs, sa mga antifungal drugs. So, mahirap. Another one is the infection caused by Talaromyces marnefii. So, this one, usually ang nagkakasakit nito, yung mga AIDS patient, yung mga may HIV. Kasi fatal systemic. So, disseminated disease. So, once nagkaroon sila niyan, ito nga, na-resolve siya. No? Nakita mo na wala na yung kanyang mga butlig-butlig ng kaunti. Pero usually, ang mga AIDS patient, yung mamamatay sila. Kasi hindi siya matitreat. Pero ang dito, ang marnefii is endemic sa Asia. So, Thailand, India, China, Hong Kong, Vietnam, and Taiwan. So, I don't know, bakit walang report sa Philippines? Meron siguro, I'm sure.
Yeah, hindi lang
But that is for you guys to know, to do. Yun, sa audience, you have to do something. Marami tayo dyan, makikita mo yan. Okay. So historically, we're going to talk about why fungi have two names. One species had two names. So historically, fungi have names for each state in its life cycle. So do you remember anamorph, teleomorph? So, minsan may name siya sa teleomorph lang, may name din ng anamorph lang because they don't occur together all the time. Minsan, teleomorph lang siya talaga, hindi nakikita yung anamorph. Minsan, anamorph lang siya, hindi nakikita yung teleomorph. Kaya yung discovery, bibigyan mo ng pangalan, yung asexual, wala kasi hindi nakikita yung sexual. So, kaya nagkaroon tuloy ng dalawang pangalan. One for the sexual and one for the asexual state. Okay, so let's go to how it came to be. So yung traditional classification of the kingdom fungi, it was based on sexual reproduction. So ang teleomorph state is the systematic state. So ibig sabihin, yun ang ginagamit for classification. Yung anamorphic state, non-systematic. So, hindi siya ginagamit for classification. Kawawa naman. Tapos, so based on this is the traditional classification before 2007 because something happened in 2007. So, based on sexual reproduction, so Chytridium-mycota, so mga chytrids, they produce flagellated zygospores. And then, yung zygomycota, nag-evolve na yung chytrids, nawala na yung flagella ng zygospores, naging zygomycota na siya. Tapos, yung ascomycota was classified based on the production of ascospores. And then, yung basidiomycota, yun ang basidiospores naman. Okay, so that's how they classified fungi traditionally. And then, let's talk about the anamorphs. There are three types of anamorphs that encompass both the ascomycota and the basidiomycota, meaning that yung basidiomycota, they produce these anamorphs too. Yung ascomycota, parehong nagpo-produce ng ganyan. But they had a superficial classification. If you've heard before, fungi imperfecti, it's not being used now, but it's still being unofficially used. Para lang merong parang holding cell or something. Para diyan mo na lang ilagay doon para meron silang ano bang tawag doon? Parang basket. Ganon. Tapon mo lang doon kasi fungi imperfecti sila. Hindi sila sexual. Okay? So also they're also called before Deuteromycota. So, there are three types of animals. Yung Blastomyces, which comprise the yeast and the yeast-like fungi. And then you have the Hyphomyces, wherein the conidia is born outside on the hyphae. And then you have the Coelomyces, wherein the conidia is born inside a sac called the conidioma. So yun ang difference nilang dalawa. Yung isa nasa labas, walang sac. Yung isa nasa loob ng isang structure. And then the Basidiomycota produces species in the Basidiomycota produce the Blastomyces and the Hypomyces. Tapos ang Ascomycota, they produce all three types of anamorphs. So yun ang mga anamorphs. They can be Basidiomycota, they can be Ascomycota. Like yeast can be Basidiomycetes or Ascomycetes. So merong mga ganyan. So you have to know what you have. Okay. I would like to reiterate. Pag sinabi mong spore sa fungi, ang tamang word, ang tamang term ay conidium. Plural conidia. Pag sinabi mong sexual, yung teleomorph spore, ascospores or bacillospores. Kasi pag sinabi mong spore, maybe you're talking about plant spores or bacterial spores. Pag sinabi mong conidia, walang duda yon, you're talking about fungi. You're talking about the fungal spore. Okay? So from now on, conidium, conidia. Okay. So the dual nomenclature. So in the old days, yung nomenclature ng fungi was under the International Code of Botanical Nomenclature or ICBN. Tapos doon sa Division 2, Rules and Recommendations, meron doon ang Article 59 that provides for the naming of each state independently and discussed in this Article 59. Kasi yun na nga nangyayari independently because sometimes you can only see the teleomorph and sometimes you can only see the anamorph. So anong gagawin mo? Hindi mo naman mahihintay na magkaroon sila pareho na play. So you have to name one, whatever you see. So yun ang nangyari. So they made provisions for that. So that's why there was dual nomenclature. So for example, Aspergillus thermomutatus, ang kanyang teleomorph, Neosartoria pseudofisheri. Parang ang layo, no? Dapat naman kahit konting pareho ng epithet man lang. When I say epithet, yun yung species name. So for example, Aspergillus thermomutatus, yung epithet niya is thermomutatus. Yung Aspergillus is the genus. Okay. So, okay, so back to that. Yeah. So why the dual names? So it says here, the same fungus can occur in many different forms. So you have the sexual and the asexual. So you have to have names for each state. And then many fungi have been described and named at different times without the anamorph teleomorph connection being recognized. So at that time, in matagal na panahon, wala pang DNA sequencing. Hindi mo ma-connect yung teleomorph anamorph connection kasi hindi mo magagawa yung sequencing. But what they did before was they did mating experiments. Kasi kaya naman siya hindi nagpo-produce ng teleomorph tsaka anamorph at the same place. Kasi merong fungi or fungus na heterothallic, meaning isa lang yung mating type niya, either mating type minus or mating type plus. So kung minus lang meron siya, wala siyang plus, hindi siya makakagawa ng sexual, ng meiosis. So ang nangyayari, kung marami kang isolates of that fungus, kung i-cross mo siya or i-mate mo siya together, so you culture them in a plate, like in a cross manner like that. And then if they have the mating plus and the mating minus, mating type plus and minus, mag-form siya ng sexual state. So most of the time, maraming heterothallic na fungus. So that's why hindi mo makikita ang teleomorph until you mate them together. Okay? So, here's an example. Yung Arachnomyces na genus. Meron siya, typified siya sa ang species niya, Arachnomyces sulphureus. So, na-discover siya in 1902. And then, when I say teleomorphic species, ibig sabihin, ang kanyang anamorphic state ay hindi known. Pag sinabi mong teleomorphic state, ibig sabihin, meron siyang anamorphic state. Sana hindi ko kayo nako-confuse. So, usually, when you read something in the article, okay, Aspergillus is a teleomorphic state. Aspergillus is a teleomorphic species. So, ibig sabihin, nagpo-produce siya ng teleomorph, pero hindi siya nagpo-produce ng anamorph. Pero mali yun. Yung example lang yun, ha? Huwag kayong maniwala sa akin. Kasi, ang Aspergillus, meron siyang teleomorph at saka anamorph. Alam yun. Example lang yun. So anyway, ang Arachnomyces na-discover siya 1902. And then in 1990, they discovered Onychocola. And this is a nail fungus. It infects the nail. Kaya, so see, this is a good example of having a name that would give you an idea what that is. Kasi Onychocola, yung infection sa nail is called onychomycosis. Kasi yung nail, onycho yun. Tapos, pag sinabi mong Onychocola, teka lang, ano Onychocola? So, it's a species of fungus that infect the nails. Okay, so that's why it's very informative. So, nangyari sa Onychocola in 1990, they had several isolates. But this was an anamorphic species. So, hindi nila alam kung ano yung teleomorph. So, ang ginawa nung supervisor ko, siya yung naka-discover nito. Ni-mate niya yung mga isolates niya. And then, it produced a teleomorph na kamukha nung Arachnomyces. So that's how they connected Onychocola to Arachnomyces. So now, because the teleomorph is known, and at that time, this is before 2007, at that time, when you know the teleomorph, you adopt the name of the teleomorph. So then the name of the anamorph gets lost. Okay? But yun na nga nagkakagulo na because clinicians like doctors, what they know is what they see. So Onychocola ang alam nilang nagkukos ng disease, hindi naman yung Arachnomyces. So kung kami mag-identify kami ng fungi and we say, hey, your fungus is Arachnomyces, like ano yan? Pero pag sinabi namin Onychocola, oo, alam namin yan. Pwede namin yan i-treat ng ketoconazole. So, ganyan ang nangyayari. That's why, naging confusing na. Ito yung, the next slide please. So, ito yung, yung example ng species na may mga dual names na magkaiba. So, ito, for example, Arachnomyces nodocytosus, Onychocola canadensis. So, ito usually ang mga anamorph names, yun yung mga clinical na name, yun yung alam ng mga doktor. So you have Neosartoria pseudofisurae, it's Aspergillus thermomutatus. Ito pa, nakakatang twist din, presumptive Naniseopsis rizii. Ang kanyang anamorph, kasi hindi alam yung pangalan, nakikita lang yung structure na Chrysosporium-like. Sinabi namin, Chrysosporium anamorph of Naniseopsis rizii. Napakahaba. So what we did was lalagyan mo na lang siya ng acronym, CA and V. So when we say CA and V, alam namin yon. Yun yung mga nagkukos ng infection sa mga reptiles. So, see, di ba, nagkakaroon na talaga ng problem. Dual names are becoming really problematic kasi hindi mo alam. Yung policy nung time na yon, nasasabihin, you use the teleomorph name, pero hindi kami nakikinig sa clinical. Kasi, why are we going to listen to that when we don't know what they are? What we're familiar with are Onychocola canadensis, Fusarium solani, kasi yun ang nagkukos ng disease eh. Yun ang nakikita namin sa mga culture namin, hindi yung mga teleomorph. So, nagkakagulo na. So, anyway, the next slide. meron ng movement for unitary nomenclature. Ibig sabihin, isahin na lang ang pangalan ng fungus. So as early as 1990s, there was a clamor to abolish dual nomenclature. So the first proposal for doing away with dual nomenclature was in the early 2000s. Nandun ako, nagkakagulo sila. I was there in 2002. I attended the International Mycological Congress, Oslo, Norway. And so this was a big congress. It happens every, I think, 4 years or 2 years. And then so at the end of the congress, there's always a meeting on fungal taxonomy kasi di-discuss nila yung mga taxonomic matter. So it was at that meeting na merong nag-propose na we want to abolish dual nomenclature. Noko, nagkagulo na kasi yung mga who's who in mycology, mga famous names. Just ko dahil akala mo magsusuntukan na sila talaga. Yung mga, hindi naman, exaggerated lang sa akin. Parang, joke it out na kasi babae, kilalaki talaga, ang grabe kung mag-insultuhan na. Kasi yung mga ayaw nila mag-change, yung mga clinicians, yung mga clinical mycologists, ay yung mga taxonomists, gusto na nilang mag-change para simple na lang kasi nakakalito na nga talaga. So anyway, nung time na yon, hindi nanalo. 80 lang ang nag-vote para sa change. There were like 500 there at that time. So then, talagang nag-gain na ng momentum. So, in 2010 or the next slide, please. So, in 2011 pala, so nagkaroon na sila ng symposium sa Utrecht in the Netherlands. So, they decided that fungus can only have one name. So they had what they call the Amsterdam Declaration on Fungal Taxonomy 2011. Para bang Treaty of Paris? Napaka.
Yeah. Sounds so official.
Mycologist na to. Sabi talaga nakakatuwa lang yung, what? Amsterdam Declaration? Parang ano yan?
Para hindi na daw po i-challenge.
Oh, so because they said that the new nomenclature of fungus or fungi is a concept that has outlived its usefulness. Pwede naman talaga kung talagang gugustuhin. So under the auspices of the International Commission on the Taxonomy of Fungi, ICTF, hindi IATF ha, ICTF. The purpose of this was to determine how the current system of naming polymorphic fungi should be maintained or changed now that molecular data are routinely available. So yung molecular data, yun talaga ang naka-cause ng change nito. Kasi yung pag sinabi mong molecular, yung DNA, yung genes, they tell you the truth. Na yun talaga ang real, tawag doon,
identity or
oo, yun talaga yung yung classification, yung natural classification talaga. Hindi yung katulad nung kanina sa magkapareho kayo ng itsura, diyan kayo classified kayo. Kasi have you heard of convergent evolution? Yun yung when you're in the same adverse condition, you tend to, react to that adverse condition. And then you tend to look like similar to each other. So yun ang nangyayari kasi merong isa parang pseudo na ascoma pero hindi naman talaga kasi kunyari lang para siya mag-survive. So yun ang nangyari, convergent evolution, yun nangyari kaya very plastic yung ang tawag nung traditional classification kasi morphological concept yung structures lang. I'm sure sa plant ganun din, maraming ding plasticity. Anyway, so next is so in the next year, so they said, okay, we will have one fungus, one name. Ngayon ang question naman, which name? Kasi madami, minsan may tatlo, may dalawa, tatlo. Excuse me. So doon pa rin sa Utrecht, sa Netherlands. So yun, pinag-usapan nila yun. So then, kung kailangan mo ng approval sa change, you have to go to the International Botanical Congress. So nangyari ito in 2012, there was a congress in Melbourne sa Australia. So na-approve yung proposal ng mga mycologists. They call it the Melbourne Code 2012. So yung Melbourne Code talaga, it was a turning point as well. So after January 1, 2013, a fungus can only have one name. So isa na lang. And then the code was changed. The name of the code was changed from the International Code of Botanical Nomenclature to the International Code of Nomenclature for Algae, Fungi, and Plants. Kasi dati kasi nasa botanical tayo. Kasi sa akala nila, ang fungi are plants. Hindi ako papayag. Hindi talaga. So then another one important thing that was changed, which I was very happy, was the language for validating diagnosis in the code can be either Latin or English. Kasi dati, pag nag-describe kami ng fungi, Latin talaga yon. Puro Latin yung description mo. So ngayon, pwede ng English, di mas madali. I had to hire somebody para ayusin yung Latin ko. Kasi hindi pwedeng hindi mo ma-publish. Okay. So again, na-change na yung code, and then what happened to that one? The language for validating diagnosis, so that is diagnosis in the code, can either be Latin or English. Okay. So let's go to the next slide, please. So, okay, what is diagnosis? To clarify the term diagnosis in this context is that it is a description that contains taxonomically informative and uninformative features, or a summary of characters that differentiate the new taxon from related or similar taxa. So yun yung tawag namin, diagnosis ang tawag namin doon sa description ng fungus. So ito yung Latin. So ganito kami magsulat. So when I published my new order and family, so I had to write it in Latin like this. And then you see my name after the name. So that's the authority of the name. And then we say ord, ordo, novum, or fam novum. Okay. So then you have to say where the article, where the name was published, because you have to register your name in the Index Fungorum, and you have to say where which page it is on. It is in. Okay. So merong ganyan. Your name will be attached to the name as the authority of that name. So kung merong mag-change diyan later on, yung name mo maging parenthesis, tapos yung kanila man ilalagay nila. Yeah.
Okay.
Okay. Kaya pa ba? Okay, so another one, the next one is an example of the English diagnosis. So this is also when I describe the species, the new species, Penicillium labradorum. O di ba? It evokes an image kasi labradorum, alam mo na kaagad, galing to sa Labrador na dog. Kasi doon namin na-isolate yung Penicillium na yan. So again, we were the authority, so my name is in there, and SP nov is called species novum. So spa nov ang tawag namin dyan. So this is the example of an English diagnosis. Okay,
so there is your name po talaga, tita. If you search it on the web, yung full description would carry your name around with it.
The name, yes.
Apo. I mean your name, the name of the scientist who coined it.
Yeah, it will. If you go to Index Fungorum, you can see our names there.
Okay.
Index Fungorum. Yeah.
So fascinating.
You're going to be a mycologist, JP. Okay, so when do you use? Kasi ngayon may English na. Dati Latin diagnosis, yung Latin lang. we just have a description, whatever we see. So now, because there's English or Latin, you can still use Latin if you want. Because you know why? Because Latin doesn't change. So kung yung description from 100 years ago, you can still understand it from today. But with English, maybe a few years from now, we won't, because some of these words will change, right? So because the English language is very dynamic, it's evolving very fast.
All the time. Yeah.
Oh, but Latin did not evolve at all. So, okay. So when to use diagnosis or description? So diagnosis, you can use that when, for example, I have a new species under the genus Aspergillus. So, for example, in the genus, there were 5 other species. So my new species is different from those 5. So yun na ang ilalagay ko sa diagnosis, what the different characters are. Pero kung ang new taxon ko or new species ko ay walang closely related na species, siya lang mag-isa, malayo siya sa iba, you can use a description. Okay, so that's the difference. You use a diagnosis, just the differential characters, para ma-differentiate mo from the others. So, palimbawa, big sila, small ka, ganun lang. Brown to, white sila. Yun. Okay, next one. Are we there yet? No, not yet. But it's clear now, right? We can only have one name for each species of fungus, whether or not it's a teleomorph or an anamorph. So now, you have to be very careful. Bad taxonomy can kill. So, pag nakakita kayo ng mushroom, ayusin nyo, identification nyo, at saka pangalan, baka mamaya, pagkain nyo, naku, either mag-hallucinate kayo kasi ang nakuha nyo clitocybe or psilocybe, yun yung mga magic mushroom. Or yung mga nagtatai kayo kasi masama, yung mayroong toxic na mushroom, yung mga amanitas. Yung katulad nitong aking drawing, amanita yan. So, minsan, meron kasi, usually, kaya bad taxonomy, meron marami siyang doppelgangers yung mga look-alikes. Na hindi naman talaga siya yung masama, pero kasi kamukha siya. Kaya you have to be very careful talaga. Okay, so navigating through the confusing changes in fungal taxonomy. Kahit kami mycologist, marami kaming nakakalito pa rin. Marami kaming nagko-complain sa amin. Ano ba yan? Bakit ba yan lagi na lang nag-change? So, the next slide, JP. So again, let's review. So the traditional classification was based on sexual reproduction, right? So then, in 2007, next one, please. In 2007, there was a phylogenetic assessment. So now we're talking molecular. So what they did here, so this was Dr. Hibbett, a very well-known mycologist, and his cohorts. So they did a very big phylogenetic assessment on all available published data, sequence data. And the results showed that many of the species, so ito yung pre-2007, nakita mo, simple lang sila, 4 na phylum, 4 lang na phyla. Nung ginawa nila ng assessment based on sequences, marami, for example, here sa glomeromycota, yung green dots, maraming lumabas na hindi pala sila related dun sa glomeromycota. So they went out of the group. Yung sa chytrids naman, merong isa, yung blastocladiomycota. So yun ang nangyari. So yung dating morphology na classification, it wasn't the true classification. Yung hindi siya natural. So ito kasi, sequences, genetics na to, natural classification. So ito na, this is what we are going to follow. Okay? And they also saw that Microsporidia, which was a a spore-forming parasite that were once considered protozoan are actually fungi. So it was very, very surprising. But Yahoo moment talaga yon. Marami kaming Yahoo moments sa fungi. You should go into mycology. I'm telling you. It's a very, very interesting thing. Last frontier para sa amin yon. Di ba? Parang sa Star Wars. Dami-dami. Ang dami-dami kasing species na hindi pa alam eh. Kaya very exciting talaga. I'm excited. Anyway, so next slide. So that was in 2007. And then in 2018, another phylogenetic assessment was done again. And then those species that were taken out of their respective groups were assessed again. And then more data came in kasi 2007, 2018, ang dami na talaga, as in sa GenBank, which is a database of sequences, we have now, as of probably December, 200 million sequences. Pero ang daming data yon, and it's for us to use. It's free. So that's a really good thing. So now, from one subkingdom, yung subkingdom Dicaria, ngayon naging 9 na. So this is now our current classification. So we have still the Dicaria, and then those that came out of the Glomeromycota, yung mga Mucorales, meron na silang sariling phylum. Meron na subkingdom, so Mucoromyceta. So naging 9 na from 1. And this was done in 2018 by Tidersu et al. Na parang European tong grupo na to. Okay. So, why were changes needed talaga? Why did we have to change? Because yung traditional classification was based mainly on morphological species concept, so yung morphology. But ngayon, because now we have demonstrated that morphology did not represent a natural grouping, then now, because of DNA sequencing, we can see that they're really divergent. For example, species A, B, and C, Penicillium, by morphology belongs to the genus Penicillium. And then, nung nag-analyze kami ng sequence variation, species B, yung sa Penicillium, went out. It's found to be phylogenetically close to Talaromyces. So, nangyari ngayon, because ngayon, doon na siya ika-classify sa Talaromyces, kailangan na niya i-change ang name niya. So, hindi na siya Penicillium, magiging Talaromyces na siya. So, yun yung pinakita ko kanina, yung Talaromyces marnifii. Bakit siya na-change from Penicillium to Talaromyces marnifii? Because of that, when they phylogenetically, when they assessed it with the sequences, nakita nila na hindi siya talaga related sa Penicillium. So, see, the genes are talking now. So, kaya karamihan na ngayon, mga gene junkies naman. Wala nang ginawa kundi mag-sequence. Hindi na tinitingnan yung fungi. That is not correct. Kasi you still have to know your fungus. Hindi pwedeng basta na lang sequence. Oh, alam ko na. A sa CB, andun na sa Talaromyces. Pero why? Do you know? Do they look the same? And they do. Kasi yung Talaromyces, para siyang Penicillium, but they produce this very, very matingkad na kulay. May red, may orange, may yellow. Yun ang characteristic ng Talaromyces. Kaya makikita mo talaga ang Talaromyces marnifii, very red siya. Meron siyang red na reverse. Ang very beautiful, pero very deadly. Very deadly, deceptive talaga. Okay, so next one is, so then why were changes needed? So the discovery of cryptic or sibling species that lack distinctive morphologic characteristics within existing phenotypic species. So here's an example. For example, yung Aspergillus fumigatus, and I'm doing this as an example kasi parang everybody probably is familiar with Aspergillus, right? So Aspergillus fumigatus, meron akong mga 50 na isolates. So out of the 50, merong 10 na katulad siya talaga. It looks the same as Aspergillus, pero hindi siya nag-grow sa 50 degrees. Pero nag-grow siya sa 10 degrees na temperature. So when they assessed the this 10 strains or 10 isolates, they found that it was really a different species. So maraming ganyan. And why was it important? Because nakita din namin na ang kanyang susceptibility to azole and echinocandins ay nabawasan. It's lesser. So resistant siya, imbes na with fumigatus, you can use azoles and echinocandins. Okay siya. Mag-work to mga antifungals na to. Pero lentulose, hindi. Refractory siya. Hindi siya susceptible. So, hindi mo siya matitreat ng azoles. Okay, so now we have to describe that as a new species because it's very important that we know what that is so that people will know or the clinicians will know how to treat it, what antifungal drugs to use. Okay. And again, as I've mentioned before in the previous slides, the same fungus can occur in many different forms, so sexual and asexual. And then many have been described and named at different times without the teleomorph-anamorph connection being recognized. Pero ngayon, without the structures itself, we can connect by using the sequencing. So ang ginawa ko, kung isi-sequence mo yung Arachnomyces at saka Onychocola na species, they should have identical sequence. Kasi pareho dapat sila na species. So that's how you can connect the teleomorph and anamorph connection without actually looking at the structures. So now, kung hindi mo ma-connect yun, magiging confusing talaga kasi magkakaroon ng dalawang names. Okay? So this is now our mantra, yun. Phylogeny outranks taxonomy every time, anytime. Talagang kung magsasalita talaga, pag papapiliin ka, phylogeny talaga is the correct one as opposed to morphology. Kasi makikita mo sa example ko ngayon, makikita nyo na why is it like that? Why is it related to that? Doesn't look like it, right? So this is my example. So Aspergillus. So after the name changes, so they said that, okay, we will have one name, one fungus, one name. So my good friend Hubka, Beat Hubka in the Czech Republic. So he in his lab, they did a very big assessment of phylogenetic assessment of Aspergillus. And you know, Aspergillus is a very specious genus, meaning there's hundreds of species, very big, like Fusarium, very specious. So what they did was they did the assessment together with closely related genera. So for example, anamorph genera like Dichotomomyces, Polypacillum, Phyllosimplex. Actually, we described the Phyllosimplex as well. And then teleomorph genera, Neocytoria, Emersella, Eurotium, Petromyces. So noong ginawa nila yung assessment, nakita nila na it's monophyletic, meaning it's under one clade or one group with one ancestor. So they said, okay, they're closely related. So now, because of the one name, one fungus, one name, we will list them under Aspergillus. But tingnan nyo naman, look at the structures of these. See, it doesn't look like an Aspergillus. So when you look at this in culture, and you don't know that there was some changes, you would still say, oh, I'm seeing a polyphyllum. No, it's not. It's Aspergillus now. So nowadays, people are getting to be educated with these new names. So that's our role as the lab, the identifying lab, to tell them that no, it's hindi na yan yung name ngayon. So for example, if I identify it as a Phyllosymplex, for example, then I would say formerly known as, so that they would know that it was named this, but now it's currently named this. So, kasi minsan, hindi naman minsan, most of the time, they will look up the name of the fungus or cases or infections para makikita nila yung mga ginagamit na antifungal drugs. So, but if they don't know which name to look up, hindi nila makikita yon. Okay. So, so now, because of that, we have a framework for going forward. So because mycology is very big, there are working groups. For example, there's working groups for Aspergillus, for Fusarium, for dermatophytes, yung mga skin fungus, yung mga tinea, yung mga, anong tawag doon sa Tagalog? Mga an-an, yung mga ringworm, yon, merong isang grupo yon. So, because it's so big, so they can focus on those groups and then they can do the taxonomy, whatever it is that they need to correct. Okay, so there's a lot of transfer of species from one genus to another and then renaming. Diyos ko, malilito ka talaga, maluloko na ka. Pag meron kaming ina-identify, then it's my job to know whether the name is current or not. Otherwise, ako yon. Ako ang responsible doon. Okay. So I'm going to give you an example how and when to describe a new species. So next one, please, JP. Okay, so I'm going to give you an example on my work. This is part of my dissertation when I was in Canada. So there was a big study in Ontario a long time ago, like before Jeff and I went to Canada. So that was in 1990. So they had this study. They were looking at 400 patients, and they got samples from atypical nails. Yung mga ganyang itsura yung sa picture. And then a lot of isolates were taken, and they were most of the isolates were identified as dermatophytes like Trichophyton or Trichophyton, Microsporum. But there were, because hundreds of isolates were done, but out of the hundreds of isolates, there were 5 that they couldn't identify. So next slide, please. So ito yung 5 isolates. Nilagay nila, dineposit nila sa UAMH, doon sa University of Alberta Microfungus Collection. And sabi ng supervisor ko, Connie, you work on these ones. I'm gonna, but it's not part of my thesis. Sabi ko, no. And so she said, but no, you work on this one as a side study. So I did. But what happened was, it was very unusual, because when you infect the nail, you're supposed to be keratin degraders, because the nail has keratin, and they were eating the nails. But when I tested them, they were not keratin degraders at all. So like, okay, ano to mga to? Are these dermatophytes or non-dermatophytes? Kasi they were looking like they are really true pathogens. Because there were other cases not only from this 5. So then I started my study, and then what I did was, so here's what it looked like. These are the anamorph states of those 5 fungi, okay, or the 5 isolates. So you can see there are different types of anamorphs. So yun pa lang, nalilito na ako, okay? Are they the same species? So ito yon. So I had to do my question when I did my proposal. So you have to present your proposal sa iyong panel. So okay, these are my questions. Are these 5 isolates the same species? Are these 5 isolates dermatophytes? Are these 5 isolates true pathogens? Yung mga ganon. Then I have to go and answer those by doing the experiments. So what I did was at that time, so I did mating studies. So in all combinations, so I kinross ko sila lahat. And then I did also DNA sequencing. So sabi ng supervisor ko, this is part of your thesis. Kung hindi pwede kasi magme-mate ako eh. Hindi ko alam kung kailan mangyayari yan. Kasi minsan mga fungus, mga ulit yan. Pagka sinabi mong mag-mate kayo, ayaw mag-mate. Tapos pag ayaw mo namang pag-matein, tsaka naman produce ng ascospores. Nakakainis talaga. So anyway, that's what I did. It almost cost me my PhD kasi 8 months na, naghintay na ako. This is my last paper, my last article to publish, and then I can have my book. Hindi pa rin, 8 months na. So I said, no, I'm gonna give up. Kailangan itapon ko na kayo. Ang dami na hundreds of plates. But, sabi ko, sige, itatapon ko na kayo today. Pero before that, I will look at you. I will look at these plates one more time. Pagtingin ko sa mga plates, after 8 months, Yahoo moment na naman. Kasi nakita ko na yung mga ascospores at saka ascomata. Natakot sila kasi autoclave ko na sila. So then,
ayaw nila titang makremate.
I know! So then, ang nangyari, when I saw those ascospores, it reminded me of Arachnomyces. Kasi these ascospores are what they look like with the Arachnomyces teleomorph. So then, na-connect na namin ngayon na meron kaming bagong anamorph sa Arachnomyces teleomorph. So what I did was, okay, but then when we did the sequencing, it was distinct from the other species. So what do? We have to describe this as a new species. So then I named it Arachnomyces cañei after the great mycologist in Canada, Dr. Julius Cain. He wrote a book on dermatophytes. And then the anamorph name was Onychocola cañei. Ang galing ko, di ba? Kasi pareho sila ng epithet. At least, hindi ako masamang mycologist. Hindi ko nililito yung mga tao. Kasi pareho sila. I mean, you can actually think right away, oh, ito yung anamorph ng Arachnomyces cañei. So, okay. So now we have a new species. We have to publish it. Pero anong lineage nito? Yung lineage, for example, genus, anong sunod? Yung family, anong family to under? Anong order to under? Anong, kung Ascomycota ba to o ganyan. So alam namin Ascomycota siya. But we don't know whether it is, which order it is in. So nung ginawa namin, nagawa na naman kami ng phylogenetic assessment. Ito yung next one. So when I did my phylogenetic assessment with this, together with most of species, like representative species in the phylum Ascomycota. So malaking tree ito. If you see that it is, so we assume that it was in the Oniginalis, but it's not. It's outside. It's its own distinct group. And then it is also sister to the Urotialis. So what do we have to do? Sabi ko, well, because if we don't erect an order and family our species will be incertiades, meaning unknown, undefined. Wala siyang place, wala siyang lugar. So, hindi pwede yon. We have to erect an order and a family. So that's how it came to be. So that we accommodate our species, so we had to erect an order and a family para merong bahay yung aming species. Okay? So the next one is, now we publish it. So we have this one, Arachnomycetalis, So Arachnomycetalis is the order. So you see, A-list is the suffix of orders. ACE or CE is the suffix of family. So Arachnomycetalis, Arachnomycetaceae, and then the authority, which is us. Okay. And then we also published the species in another article with the span of, with my supervisor and my panel. Okay. So next one, please. So since I became a mycologist in 1998 until last month, I have discovered 16 species of fungi, and I have erected or established one order and one family. Okay, next one. So for you guys, if you have names of fungi, if you want to check whether the name of the fungi is still current, you can go to these websites and check from there. And also it's very good to do to look at publications, because usually the publications will be very, very up to date. Sometimes Index Fungorum is not up to date because the people who are working there are only voluntary. So kung kailan lang sila may time. Okay. And then also, if you want to look at the next one, JP, look at names of Penicillium and Aspergillus, you can go to this website because the names there will be correct. Because there are many names now that are not Aspergillus anymore. So if you want to be sure, go to this website and they're always updated. So here is Index Fungorum. So as you can see, the name listed in blue, for example, Neosartoria fisheri, is the old name, and the one in green is the current name. So that's how you check for the current names. Okay? So now, is it the lab's role to report name changes to the clinicians? Absolutely. It is our role to do that because we are the ones who are studying them. We are the ones who are looking up these things. So it is our responsibility. Thank you.
Ayun.
All right.
Ano ko. Maraming. There's another slide after this, actually. Okay, so before po tayo pumasok sa Q&A, Tita, merong nakisali sa webinar mo. Hi, Taho. Taho, say hi to everyone. Okay, so I would actually I'm delighted to announce din po that Tita Connie actually suggested, and that we have follow up webinar workshops po. Kasi and Tita Connie, I would like for us to be able to chat about this now. Ano po na before we actually schedule this, Tita and I nagkumustahan po kami, and we talked about Felsyab, and she actually mentioned for the lack of better term, frustration, kasi po sabi ni Tita, the Philippines is actually biodiverse when it comes to fungi. But unfortunately, most of the most recent discoveries of new species in the Philippines were actually done or reported by foreign researchers. So what we're actually hoping to do is to also, share Tita Connie's expertise rin to influence younger generation of Filipino STEM enthusiasts who would actually like to venture into mycology and taxonomy research. And so we have a couple of follow-up webinars po that wala pa pong schedule, but we will be announcing quite soon over the next few months. So the first of which will be an update on the identification of fungal diseases. So perspective from a human international clinical reference laboratory. So, I think this is actually currently what Tita is doing right now. She's part of a medical research institution in San Antonio, Texas. And so I think this is actually going to be very exciting. So please spread the word po because this is a very rare opportunity for a world-class Filipino to actually share, like some of the things that she learned from the international research scene, like more particularly clinical facing. The second naman po is actually on biodiversity of fungi. So ito parang workshop to, no. Hopefully yung mga bata lalo maka-attend nito. So she'll talk about isolation, identification, and preservation of fungi. And then very lastly, if you can read the one-liner at the bottom. So if you're interested in collaborating on Philippine-based fungal discovery research, this is something that we're trying to come up with the mechanism through which of teachers and students in the Philippines who would like to venture into this. Can you hear me, JP?
Yep.
Yep. There could be groups of teachers and students from the Philippines who are taking an interest in fungi discovery research. So we'll try to come up with a mechanism para po ma-mentor kayo ni Tita Connie. But then ito lang po for I'm medyo disclaimer lang, since gusto po talaga namin that like we are really fostering the culture of research among Filipino science and teachers. Hindi po namin kayo i-connecta kay Tita Connie kung ang tanong niyo po, bigyan niyo po kami ng topic. Come up with a topic, express your intention, and then if your intentions are actually kung makakapasa kayo sa audition kasi kami yung magsi-screen, we will hook you up with Tita Connie. So that's actually one thing that we're glad to announce. So watch out for some announcements po on our Facebook platform and on our website kung paano po ba natin magagawa ng mekanismo ito. So before pa mag-Q&A, I would like to thank Tita Connie because I think this thing that we are initiating through the Felsyab platform, it's a non-traditional way po of connecting to the Filipino educational sector. Kasi if our high school students are actually made familiar with like the opportunities in as career fields, and mas marami po siguro yung may encourage to actually follow in like the footsteps of world-class Filipino mycologists like you, Tita. So yeah, thank you so much po. And then yeah, JP, we can actually move on. Ang daming tanong bigla sa Zoom. And then I was also during your talk, Tita, I was collecting some questions from YouTube. So ayun. So JP,
yeah, and I also would like to mention that dito sa last slide natin,
yep,
this is also an open invitation to all experts in taxonomy and microbiology in general.
Yep.
Marami po tayo niyan sa UPLB to come forward and help us kasi we are like embarking on a very interesting journey.
Yep, and as we said po, like what we do in Felsyab is train them in research fundamentals, but then again, yung continuity po nun lies in we can foster the sustainability of the passion of our students and teachers if there's sustained mentorship. So then syempre kami po,
and continuity. Yeah.
And we're not experts in this area, but
exactly.
we can use the platform po to connect both communities, our Pinoy scientists and experts po with with our education sector. So kung sino man po yung may interest, what Felsyab can do is to match you up, But again, we highly encourage you to come up with ideas. Your idea doesn't have to be perfect. It doesn't have to be streamlined. But I think for as long as we see the intention from your end, and hindi naman po ito yung parang hihingan nyo na resources na bibigyan kayo ng, ng lab equipment, but mentorship po and feedback. That I think those are especially from experts, I mean, those are things that, you know, hindi po siya madaling mahanap. Mas madali pong hanapin ang equipment kung may pambayad kayo, but then the expertise and mentorship, yun po yung medyo mas mahirap hanapin. Okay. So
Alright.
actually, JP, let me share my screen first, because before tayo pumunta sa Q&A, I would just like to announce a couple of events po para makapahinga naman din ng konti ang throat ni Tita, kasi she talked for an hour and a half. So after this po, next week we have some webinar workshop on data science. So yung mga high school and college students who are looking for research topics, you know, during the school year, hindi kayo makapag-lab, this workshop is actually for you, and it will be presented by one of the youngest experts in data science in the Philippines, Professor Kevin Anthony Season from St. Luke's Medical Center. So magaling po siya. And then after that, like on April 10th, we have a chemometrics webinar to be delivered by Professor Gerard Dumangcas from Louisiana State University in Alexandria, in Louisiana. So it's on April 10th. And then on April 17th po, that's the 5th Felsyab Research University training course, and we will be delivering, you know, like a lecture on basic statistical design of experiments. So ito po talaga yung hinihingi rin ng marami nating followers who actually are looking for resources and, you know, learning materials for that they can use for research. It will be delivered by Professor Lara Paul Ibal from the Institute of Statistics from UP Los Baños. And then on April 24th po, we do have a special topic webinar on cancer genome analysis. So this is to be delivered by highly reputable scientist from the Baylor College of Medicine. His name is Professor Chad Creighton. He's based here in Houston, Texas too. So that's on April 24th. And then lastly, para sa mga students and teachers po who are planning to participate in Felsyab's competition, Felsyab Research University's Lions Den's deadline for submission of proposals is actually on April 30, 2021. So dyan po, mananalo kayo ng 10,000 pesos, yung pinakamagandang idea. And then meron po tayong mga Lions choice. So ito po. So all the details of all these events are posted on our website and on our Facebook platform. So yun po, punta lang po kayo doon. And then now we can actually proceed on to the Q&A.
Yes. And siguro first come first served basis na lang po tayo dito, no? We have several questions here on Zoom chat from Larry Larioza. I wonder if Larry would like to address his question personally live. Is Larry here? Okay.
Okay, baka shy siya.
Okay. Well, he was asking that, as a background, some bacteria are alarmingly, probably dangerous, especially those species that possess flesh-eating bacteria characteristics. For example, rhinocerebral mucormycosis. So his question was, what are the possible challenges if we meet an unknown bacteria that may give threat to our health?
Siguro ano natin yung context no, baka on the context dito of fungi siguro.
Yeah.
Yeah. So yung flesh-eating bacteria siguro, Tita. What if meron po talagang ganyan, if people encounter it, and siguro how do we approach, you know, like how do we manage them?
Well, pero hindi ako nagtatrabaho sa flesh-eating bacteria. Right?
Mm-hmm.
So we deal with fungi. So when we have that kind of thing, like when we see, for example, our clients or the clinicians would send us an isolate from the patient with that cause mucormycosis or rhinocerebral mucormycosis, that will be an emergency for us. So we have to do the identification right away. It'll be like stat, because you saw the slide. After 12 hours, necrotic na. So it has to be identified right away so they can know how to do it, or what antifungal drugs to use.
Yep. And siguro, Tita, I would just like I'm gonna follow it up now. So for example, for college students and teachers, or even at the high school level, if they actually venture into fungi research, Tita, no? So how do they? I mean, is there a fairly safe system by which they can isolate things without really exposing
Yeah.
them to these
Because
things?
needless to say, parang
Baka
expert
dito
job to eh.
baka pumunta sila dito ng gubat, makaapak sila.
Yun nga.
You need to have at least a BLS2.
What's a BLS2 po, Tita?
Oh, okay. A biosafety cabinet, class 2,
Okay.
because that is something that is safe for yung ganyan ka, hindi masyadong pathogenic, pero din,
Apo.
nakaka-infect din.
Okay.
So,
So
kailangan mo ng biosafety cabinet. So, hindi ka pwedeng basta-basta na lang mag-isolate without being very careful. So, you have to not be very cavalier about it.
So Tita, for example, if we have high school students who would like to get involved in research like this, are equipment like BLS2 accessible say sa mga provincial colleges? May mga ganun po ba usually? Or is it something that's very difficult to actually find?
It's very difficult because even at UPLB, hindi ko alam, meron na yata sila, pero it took a long time to get because it's very expensive.
Okay. So siguro for things like this po, it's very important to also have like the linkage with
And also I would like to, if a student is working on that, like isolation, they should have meron talaga silang parang mentor na to guide them on the safety aspect of the isolation.
So there should be training, but okay. So again, so I think like for a warning, reminder lang sa mga tao, wag kayong basta-basta then magpunta out there.
Yeah.
Mag-isolate ng fungi. So at least like get some guidance from expert. But,
Yeah.
But then Tita, it's perhaps siguro it's like equipment is very important, but it's also not to say that it's impossible, ano po.
No, it's not impossible. Yeah. And then basic equipment, like you have to have a microscope, a dissecting scope, at saka incubator.
Apo.
Yeah. Pero usually naman kung environmental ang iyong isolation, ambient lang hindi kailangan. Pero kung clinical, kailangan mo ng 30 degrees.
Okay, so for example dito, ito just to put into context, no. For example, nasa likod bahay ko lang at nakikita ko meron diyang fungi. Is it safe to do it? To take that and like try to identify that?
Oo, basta make another parang bahay-bahay dun sa likod. Huwag kang magtrabaho sa loob ng bahay.
Sa loob ng bahay.
Okay. That's
or at least parang mag-training ka yung how to work with fungi. I mean, maybe that's one of our courses, like how to work with fungi safely.
Okay. Ayun, siguro po, isa yun sa mga follow-ups natin. So, kung mga estudyante dyan who would take an interest in this kind of research, yun yung isusunod natin na workshop from Tita Connie.
Hindi yung basta, sisinghutin mo yung plate. Hindi pwede yun.
Yeah, actually, Tita, this, like, thanks for the lecture because it brought me back to my Bio 1 and 2 and Microbiology 1 years, like, days in UPLB, but I just realized I don't remember anything anymore. But
yeah, me too.
I think there's a reason why naging chemist po ako. But then, okay, so ito I noticed this question from YouTube, no, Tita. I think this is from Kyla Corsega, and she asked, ano pong taxonomic initiative status sa Philippines? And parang nag-boom na po kasi ang mushroom cultivation agribusiness. I don't know if you can make.
I don't really know because hindi ko talaga alam kasi wala akong masyadong nakikitang publications on that from the Philippines.
Yan, kaya nga sinasabi ni Tita, maging interested ang mga tao sa fungal research. Yeah.
And if we have local taxonomists here present, so kindly come forward po to enlighten us on the current situation here in the Philippines.
Director Marian, are you still there? Director De Leon.
Tita Cons.
Yeah.
Yes. there is already an increased number of researchers working on fungal taxonomy in the country, isolating samples not only from environmental samples but also clinical samples. But of course, we still have to have experts like Dr. Gibas to teach us on the proper fungal taxonomy. But I think I heard about the biosafety cabinets. The University of the Philippines in Los Baños, meron na pong mga laboratories, tita, na marami na pong may biosafety cabinet.
Oh, that's
So,
great. Yeah.
yes po. Because, of course, kailangan po we work on using biosafety practices kasi we are also working with pathogenic microorganisms.
Yes.
And
Alright.
Okay, we would just like to acknowledge Dr. Marian De Leon, who the one who just spoke. I believe Dr. Marian is from UPLB Museum of Natural History, if I'm right.
She's the director.
Oh, okay. I'm sorry, ma'am. Ayan, so we're very delighted to have you here.
thank you
Dr.
very
Marian
much.
De Leon.
Thank you
Okay.
very much.
Ayun. And then,
So, ayan.
other questions from Zoom, JP?
From Zoom, from Crystalline Makua. What are some recent advances in collecting fungi in pristine areas or woodlands aside from the one you mentioned?
Advanced.
And
Like how to?
recent advances daw po in collecting fungi sa collection.
Actually, yung kung collection lang ng fungi from the woods, basic lang yon talaga.
Opo.
Basic lang. So
So,
you
yung
just
conventional lang.
yeah, for example, it depends what kind of fungi you want to isolate. For example, you want to isolate mga yung nasa ilalim sa loob ng trees. So mangunguha ka ng cookies, yung isang branch ng tree, kakat up mo into cookies, tapos doon ka mag-isolate. Lalagay mo
Okay.
sa ano. So it
Is that
depends.
how you call it, tita? Yung portions, cookies?
Cookies.
Oo,
Wow,
wag niyo
it's
pong
a very,
kakainin yun,
very
ano
cute
po.
term.
Kahoy yun.
Huwag niyo
Kahoy.
pong kakainin yung cookies na yun, ano po.
Yeah.
At
So,
FYI
yeah.
lang, disclaimer.
So if
Yes.
you want to isolate like yung mga phyloplane fungi sa mga leaves. So for example, you want to isolate Blastomyces yeast. So ang gagawin mo, yung sinabi ko kay Jeff dati, yung plate mo, yung agar plate mo, so may agar sa ilalim, pero yung lid mo, lalagyan mo ng petroleum jelly. Doon mo ilalagay yung iyong leaf. Para kung yung Blastomyces na yeast, kasi yung Blastomyces, yun yung tinuthrow nila yung mga spores nila, yung canidia nila. So para maging
Ma-contain.
pure yung iyong isolation, hindi mo na kailangan ilagay yung leaf sa agar kasi marami pang kasama yon. Kung yun lang gusto mo na yeast, ang i-shoot nila yung spore, pupunta na kaagad sa agar, tutubo yun ng pure culture. Yung mga ganang klase.
Okay,
Para ma-isolate na siya right away.
Marami talaga. Ha?
Medyo doable yung tita, no?
Yeah.
Okay. So, eto tita, ito next naman siguro ito icebreaker to, no? This was asked by Edward Jan Zapata. What he asked was, kung kikidlat po ba, may mushroom po ba natutubo?
Ha?
Kung kikidlat daw po ba, may mushroom daw po bang tutubo?
Di ba yun ang kwento kutsero?
Okay, so ano po ba yan, tita? Ano po ba yan?
Anong phenomenon ba yan?
Opo, totoo po ba yan o hindi? Hindi. Okay yan.
Kaya tumutubo ang mushroom kasi 'di ba umulan?
Apo.
Nabasa sila.
All right. And then there's another question po. This is from Andre American. Siguro ito po tita na curiosity question. How many species are there of fungi that cause necrosis? So meron ba tayong estimate ng ganun tita? Wala.
There are many maybe necrosis in terms of yung yung katulad ng pinakita ko.
Flesh eating?
Marami yon. Yung mga mucorales, kumakain yun ng face. Yung Cladophylophora, siguro maunti lang kasi 300 lang yung known na pathogens eh. So siguro mga ano lang, 10, 20.
Okay, so
Pero
parang,
hindi ko alam. Ang alam ko, kaunti lang.
okay. So parang talagang minalas ka na lang talaga pag tinamaan.
Pinalas ka na lang talaga.
Opo. Ayun.
Kaya minsan, if you notice, kung sa bahay nyo may mga nanonotice kayo ng mga mold na itim, mag-ingat kayo kasi baka Stachybotrys yon. Yun ang nagkukos ng sick house syndrome. Yung hindi siya tumutubo sa body mo, pero nagpo-produce yun ng VOCs, yung volatile compounds, na toxic. So, masisinghot mo yun. Kaya yung buong bahay na nakatira sa bahay, magkakaroon ng parang asthma, allergy, kasi meron silang Stachybotrys sa likod ng mga walls nila.
Okay.
Oh,
So parang
Kaya-
secondary yung effect niya.
Oo, VOCs
Mhm.
lang, yung
Mhm.
volatile compounds.
Okay. Now from Alberto Reliora, he wants to ask if there's a specific method in fungal DNA extraction. So how is this done po?
Wala. Para ka lang
Mhm.
nag-i-extract ng plant.
Okay. Ordinary DNA extraction lang.
Oo.
Okay. Is it possible to alter the DNA of a flesh-eating fungi that causes disease or to develop a specific cure?
I don't
So,
know. You
so
should
parang
ask
ano na siya.
for people.
Medyo hypothetical yung question. Yeah.
Maybe. I don't know. I'm not a gene person.
Mhm. Okay. So, we have Julie Isa Mandap. I don't know if she's here, if she wants to come forward. She wants to thank you for hello.
Hello. Yes, I'm
Hello,
here.
Julie. Would you like to ask it live?
Sure po. Sige po.
Okay.
Dr. Good morning. Thank you so much for the very exhaustive talk. Ang dami ko pong natutunan. So I am a pathologist po in UPLB and I teach general mycology. And since I'm a plant pathologist po, I deal mostly with fungal phytopathogens. So I wanted to know your insights, ma'am. If there were also fungi that you work on humans so far that can't be isolated in pure culture, in artificial culture for being obligate parasites po. If yes po, can you share some experiences po on how you handled the classification of such fungal species phenotypically and phylogenetically?
Ang experience ko, yung mahirap talaga i-isolate yung Lacasia loboy, but I haven't seen it. So, hindi ko alam yon. Pero yung isa pa, yung Malassezia, alam mo yung Malassezia?
Malassezia for fur?
It's also very difficult to isolate kasi kailangan yun ng lipids. Tapos, kasi nagkukos yun ng di ba, yung seborrheic dermatitis. So yun, ang isang experience ko na nahihirapan ako mag-isolate.
Pag ganun po ba, sorry po, may follow-up question ako.
No worries.
Pag nahirapan po kayo mag-isolate of such fungus, nag-optimize pa po ba kayo ng laboratory conditions so that you can mimic yung conditions talaga na magpo-produce sila ng tamang...
Kasi katulad noon, merong nag-collaborate sa akin. Tapos pinadala nila yung isolate sa akin. Hindi ko alam kung ano yon. So, hindi siya tumubo sa 30. Tapos tinanong ko, sabi ko, ano ba tong isolate na to? Saan to galing? Sabi niya, sa aquarium. So, nilagay ko sa 9 degrees. Tsaka pa lang siya tumubo. So, may mga ganon. Talagang iisipin mo kung anong environment ng fungus na yon. Kung siya ay galing sa clinical, then 37, 30, mga ganon. Pero kung environmental, depende kung saan yon. Kung anong host, yun din. So, kung limbawa, sa aquarium pala, so malamig yung tubig. Sabi ko, anong temperature ng aquarium nyo? 9 degrees. So, nilagay ko lang siya sa ref. Tumubo na siya. Yung mga ganon. Tapos minsan ayaw naman talagang tumubo, pero alam nila na nandun. For example, yung Exophyla, black yeast. Nakita nila sa tissue, pero ayaw niyang tumubo. Kailangan kong ma-identify. So, ginawa namin, merong yung process na galing sa yung histopath, nagawa sila ng parafilm. paraffin embedded na tissue. So doon kami nag-extract ng DNA. Hit and miss pero nakakuha kami. So we were able to identify kung anong exophyla yon. So mga ganong klase. Kung hindi siya tumutubo, diretso na, direct sequencing ang ginagawa namin. O di kaya probes.
I see. Okay po. And how
Oo.
long, kaso na lang po, how long po kadalasan, gaano katagal nyo ino-optimize yung ganong conditions? Yung based on your experience po, months? Years?
Hindi naman kasi pagka-clinical stat yun Sasabihin mo ang turnaround time, ang tawag sa amin ng turnaround time yung pagka nakuha nila yung result. Ang turnaround time sa fungus ay 3 weeks. Pero kami, spoiled ang aming mga clients kaya wala pang 1 week tumatawag na yan sila. Kaya ang ginagawa namin, kung may sequence kami, tapos isasabay na namin so lahat ng kung gusto naming mag-fruit siya, makakita kami ng teleomorph or anamorph, pipili kami ng medium. Yung halimbawa oatmeal agar, nag-i-encourage yun na mag-produce ng teleomorph. O di kaya lalagyan namin so dati gumagamit sila ng carnation leaf. Ngayon wala na akong makukuha ang carnation leaf. Ang ginawa ko, kumuha ako ng dahon ng saging. Nilagay ko dun sa agar. Nag-work. O ngayon, gusto ng supervisor ko, write it up as a procedure. Parang, okay. Parang ganun. So you have to think of things like that na kung ano yung environment, i-mimic mo na lang.
Thank you so much po, Dr. Connie.
You're welcome.
Wow, that's a very nice insight. We have from YouTube, from Alma Segismundo, our research on the antifungal activity of an indigenous plant species used to cure oral candidiosis showed a strong inhibitory activity to C. albicans. What would you suggest to be the next thing to do?
Tingnan nyo kung pwede siyang, kasi merong emerging na yeast na species na pathogenic, yung Candida auris. So, kung makakuha kayo ng isolate na yon, iti-test nyo, that would be a good thing. Kasi, parang nagiging na siya, meron na dito sa Amerika, galing yan sa Japan. Meron dito sa Amerika, meron sa Latin America, meron sa Europe. So, dito, may memo kami last week, nagiging reportable na siya. If you see something na may Candida auris, i-report mo yun sa health, sa City Health. So, kung pwede niyong i-test niya sa Candida auris, that would be a really good thing.
All right. So ito, medyo sensitive siya. With regards to newly discovered species ng yeast, are they suitable for baking or for fermentation? Mas mabilis po ba pag ferment kaysa sa common baking yeast? So let's be very cautious about like newly discovered, because as Dr. Connie mentioned, medyo dangerous yung repercussions. Kaya
hindi naman alam kung ano, dapat hindi mo alam kung yun pwedeng gagamitin sa baking, di ba?
Yeah. Kasi nabanggit lang kasi na yeast, baka right away gamitin na. That's why taxonomy and identification is really so important.
Para po yung mushroom, hindi po lahat ng mushroom pwedeng kainin. Gaya na sinabi ni kanina. So, sabi nga ni Tita, you have to know your fungi.
Maraming doppelgangers Marami talaga.
Ayan. Ito comment lang po from Jezreel Paulite from YouTube. Wow, the last time that I heard the voice of Dr. Connie was during the exam of Ma'am Fe Sulaybar's necrological Zoom meeting. So parang very siguro, I don't know how long that was, but
last
he's
week.
probably sending his regards.
Kasi Fe passed away.
Yeah. That's sad. Yeah. Ayun, meron pa ba tayo sa Zoom?
I just have one more. From Julie Garcia from YouTube. Do fungi perform bioremediation po like bacteria?
I think so.
Okay.
Ginagamit din siya.
So yeah, magandang ano din po yan. Magandang
Marami
area.
talagang fungi eh.
Opo.
Kasi most pathogens, most yung nagkukos ng infection, mga opportunistic. So because marami na kasi tayo ngayon may mga sakit-sakit. So yun ang anuhan mo. Dapat yun usually yung nakikita namin na mga isolates are from transplant patients, yung may mga diabetes, may mga cancer, HIV. So yun, sila dapat na natatamaan talaga sila ng fungi infection. And you know how difficult fungi infection is to cure. Kasi yung antifungal drugs ay harmful din sa atin. Kasi eukaryotes ang fungi, di ba? So tayo eukaryotes din. So what harms the fungus also harms us. So kaya ang ginagawa nga pag nag-treat ng fungi, very dilute yung it takes months bago nila matapos yung treatment. Kasi unti-unti tapos yung binomonitor yung drug levels sa blood. Kasi pag tumaas yung drug levels, very toxic yon. Yeah.
Okay.
All right.
So I guess we can conclude the Q&A for now.
Yep.
If you have questions, kindly put them in the evaluation form, which I will roll out later.
Okay.
Okay.
So okay, punta lang po kayo doon. So ilalagay namin yung link doon sa YouTube description box, and also go to our website later kasi ipo-post po namin yung kung paano yung pagkuha niyo ng e-certificate. But before we all leave, baka po pwedeng mag-request tayo, mag-picture tayo with Tita Connie para meron tayong remembrance with Tita. So for those of you po who are open to turn to activating your cameras, please.
Last picture po tayo.
Last picture tayo.
If you wish lang po. Optional po ito.
Kung comfortable po kayo. All right. On the count of 3, 1, 2, 3. Okay, one more. Kasi apat po siyang
Ay,
panel.
stop!
1, 2, 3.
Ana!
Ayun. And the Tita, are you seeing lots of familiar faces po?
Yeah!
Yeah.
Naman!
We will send.
To have a reunion.
Are you the only one? Thank you.
Ayun
Hello,
po.
Ma'am Sok. You can unmute po.
Dr. Sok. Oh, Dr. Ana.
Ay, isa pa po kasi yung iba po hindi naka-activate yung camera kanina. Okay, one more. 1, 2, 3. Ayun. Maraming salamat po. Ayun.
Thank you.
Tita Connie, maraming salamat po.
Before anything else, I would like to present Dr. Connie our certificate of appreciation. Okay, Felsyab, in behalf of Dr. Jeff Bunkin and the rest of the team, we would like to present this certificate of appreciation to Dr. Connie Cañete-Gibas for delivering the webinar, What's New in the World of Fungi, Taxonomy and Nomenclature on the 27th day of March 2021, signed by Dr. Jeffrey Bunkin, CEO and founder of Filipino Science Hub, and myself, JP Onya, Head of Research, Filipino Science Hub. So maraming salamat po, Ma'am Connie, for this very insightful webinar. Ayan.
And then like we said, marami pa pong kasunod. May dalawa na tayong nakalinya po, and then si Tita po, Tita Connie, we consider her part of the Felsyab family already. So marami pa po kayong aabangan kay Tita.
Sana may natutunan kayo.
Ay, Tita,
very thankful po ang ating mga participants from Zoom and also from YouTube. We have we are full house here on Zoom.
I think that on YouTube, like, we peaked at about like 150.
And we have 100 more.
Yeah, so 250 participants, Tita.
Yeah,
so you reach.
Nakapuno na tayo ng lecture hall.
Yeah, nakapuno tayo ng lecture hall. Yan, Tita, maraming salamat po.
Maraming salamat po. And our participants come from all over the country. We have people from up north, Abra, Pampanga, and also from Silliman University, yung schoolmates po nyo are also present. So maraming salamat po sa pag-participate po this morning.
Yeah.
And I will share the link for the certificate later. Okay?
Okay,
So,
punta
do
po
you have
kayo
any
sa
other-
website. Nandun po yung link. Give us siguro po mga 20 minutes. We will upload the guide po. Filsyab.com.
Do you have any other?
I think we have already advertised like our next event, so I think we're good. Maraming
Okay.
salamat po uli sa inyo. Tita Connie, I'll see you soon in a month. So, okay, maraming salamat po uli.
Kasama ka, JP.
I wish po, tita.
Opo. Next time,
Okay.
tita, next year, we'll have a get together.
Wait, I'll
Somewhere.
just share the Google form link here. Ayan, hindi ko na po na ano. Sorry po. Form for the link. Ang hirap mag-multitask.
It's okay. All right. And I also uploaded the guide to get the certificate like on the website. So you guys
Okay.
can also check it out even if we end the session. Maraming salamat po.
Ayan, maraming salamat po.
Thank you,
Sa
everyone.
mga participants po natin from YouTube, kindly check the live chat right now for the form, for the evaluation form, because hindi po namin may issue yung certificate if you don't accomplish this form. So yung registration po is for the Zoom. So not necessarily makakakuha po tayo ng certificate from registration. So we have to accomplish this form, and kindly wait for 1 week to receive your certificates, because we are giving out certificates in batches. So kung hindi po dadating bukas, baka sa isang araw, ayun po. Kindly follow up po from us your certificates after 1 week when we have completed the issuance.
Yep.
Okay?
And make sure po tama yung spelling ng email at information.
Tamang spelling ng name, and kindly double check your email address, kasi magba-bounce back lang po yan kung mali po yung email address. Alright? Okay.
Right. Maraming
With that,
salamat.
maraming salamat po. Papasalamatan po ulit natin ang ating guest speaker, Dr. Connie Cañete-Gibas from Texas, USA. So I hope this is the first of many web offerings with Dr. Connie. So maraming salamat po. I think Dr. Socorro Parco is sending her love and appreciation to Dr. Connie. Maraming salamat po, and I guess at this point we can conclude. So pakiantabayan na lang po yung mga next announcement from Felsyab from all of our channels. So maraming salamat po. It's already 12:30 and 12:30 na po in the Philippines. So magtatanghalian na yung mga tao. Okay. May you have a very nice weekend. Happy lunch, and we'll see you next time. Actually, next week. Next week we have another offering about data science. So kindly watch out for that.
Okay. Tita, thank you.
Thank you po.
Have a good night.
Yeah,
Maraming salamat po. Ingat po kayo lahat. Okay.
Opo.
Bye.
Thank you.
Bye-bye.
Thank you, Marian. Bye.
Hi, Tita Con. See you.
Bye. See you someday.
Someday. All right.